All right. Hey, Tom.
How you doing, David?
Doing well. How you doing?
Very good.
Thanks for joining us this morning.
So,
Um, yeah. So, and thanks to everyone for carving out time and your out of your busy schedule and happy holidays. Uh, yeah. Let me just start out with some opening remarks. So I'm David Fleck, founder and one of the managing partners at Freeflow, and today really pumped to have Tom Miller join us. So Tom is the co-founder and CEO of Iambic Therapeutics, and Iambic is actually the first investment of our firm. Back in April, I believe, of 2020 is when that was consummated. So in the time machine, that people probably can go back in time and realize that that was a fairly chaotic time, and that's part of the journey that we've had together. And when we launched the firm, we saw a very interesting opportunity to focus on deep tech enabled by compute. What we call CS plus X, essentially because in many ways that's what Caltech has called it for many years, actually before us. Computer science plus chemistry, computer science plus physics. Just like what can you do by coupling computer science with other scientific disciplines? And that's been the foundation of our firm. And when we invested in Iambic, it was just a natural because at the time it was basically, I'm not even sure if you called it AI drug discovery at the time, but it was, almost science fiction to think about what you could do by using compute for drug discovery. And that's been something that people have talked about and thought about for a very long time. And I think in some ways there was hype cycles around it and less substance sometimes. And I'm happy to report that Iambic is proving that with material clinical assets and partnerships and things like that that Tom I'm sure will come out in our conversation is now just validating that you can actually do this. And I think has built a very interesting company that is powered by two engines, its own assets, and then also it's working with large biopharmas who have realized that they almost have to work with someone like an Iambic. To the way, don't want to put too many spoilers in here, but very, very honored to have you join us today. And let me pass it over to Tom for some opening thoughts and as well as to just kind of describe the company for those who are less familiar with Iambic.
Happy to do that, David. Thanks so much for having me on this opportunity to talk. Great to see you as well, Kevin. This journey with Freeflow has just been a fantastic one, as you know, from the earliest days, and I'll be excited to highlight that and to tell you, where we've come and where we're going as a company. I'll I'll bring up a couple of slides, and then we can jump into it. I'm excited there'll be some chance for question and answer at the end. So anybody who has questions along the way, I think they can just probably drop those into the chat for David, and we'll look forward to digging into those. At a really high level, Iambic does three things. We focus on technology and AI innovation. This includes two flagship tools, one for three-dimensional structure prediction, one for broader preclinical and clinical property prediction called Enchant. I'll be giving you a little bit more of detail about both of those. But we're not just an AI company. What we do is we seek to take those algorithms, those methodologies, and we combine those with wet lab automation so that we can go from new designs to chemical synthesis to biological testing and closed loop discovery cycles on the fastest possible pace that we can. And that's core to how we execute our drug discovery cycles and projects here at Iambic. And, really, as David pointed out, there's a lot of excitement about AI. There's no shortage of AI for drug discovery companies, but actual companies that have executed to clinic and through clinic are far more rare. And really showing that these things can move the needle in terms of the probability of success of programs, the quality of assets that are created, the pace to getting to clinic and to patients is really a critically important measure of success that we try to hold ourselves to at Iambic. At a high level, Iambic is over 100 people now. We've got a sizable machine learning and software team, a sizable in-house chemistry and biology team, a growing clinical and G&A team. We've raised over $300 million at this point. This includes an oversubscribed $100 million round that we announced about a week or two ago. But I want to talk about the first $3 million, the seed round, and that's what we got in 2000. And David and Kevin, they were part of that seed round. They helped us to nucleate that. They helped us to do that in COVID. And that was a hard time. We were inexperienced PIs at the time. Well, we were experienced PIs. We were inexperienced entrepreneurs. It was just a fascinating time and process to launch this company back at end of 2019, start of 2020. And Freeflow was a great partner in that journey. And they've stayed a big partner in that journey. I highlight to say they've continued to be strong supporters throughout the subsequent years and subsequent rounds. And that's been an exciting process. In 2020, I mean, I don't normally emphasize this in my short talks, but when we initially launched this company, we did not know we would be a drug company. We did not know that we were going to use the underlying chemistry and software and AI platform for that particular application. We were open to the possibility that it could be polymers or that it could be catalysts or that it could be, other applications. And it was, drug discovery that turned out to be the right fit at the right time with the right journey towards productization. And we leaned into that. But that meant that in the first year or so, we really focused on proving that underlying chemistry-based technology. Then, once we had that more clear line of sight, we did a Series A that allowed us to build in-house laboratories here in San Diego. We launched internal programs. We've made the journey to clinic in less than 2 years with our first program and with additional programs on the way. And even in clinic, we've now gotten to the point where we have proof of concept data in patients that have now disclosed publicly as well and continue that journey. So it's been a very exciting process, and it's equally exciting that there's a lot more that we stand to do as a company and look forward to doing that. Really focusing on the impact of AI for patients and really the realization of the promise of AI is something that I like to emphasize at Iambic. Again, many drug discovery companies, but if you take the compilation, as in this case from the Boston Consulting Group, of the examples of AI drugs that have actually made it to clinic and how long that took, Iambic stands up really compellingly in that journey. Less than 2 years from program launch to our first clinical entry is years faster than the industry average and years faster than the next fastest examples. And not only that, we've not just gotten to clinic, but we are, as I mentioned, this fall reported our first clinical efficacy and safety and data at the European Molecular Oncology Conference. That was, I think, another major milestone in that journey, showing that not only can we get to clinic, but we can show differentiated data in patients at that time. And we're excited about the continued progress for that with our first potential approvals as early as 2028 and a clear line of sight towards that as well. The way we do this at Iambic is with an in-house platform. That platform recognizes that there's many things that you have to get right to go from a target of interest to a small molecule drug that's actually going to succeed in clinic. And, this is a process that's been run by many folks over many projects over many years. So there's a lot of heterogeneous multimodal data for many different endpoints that is available. What we do at Iambic is we take that data, we fold that into foundation models, two flagships of which are Neuroplexer for 3D structure prediction and Enchant for basically everything else, all the other preclinical and even early stage clinical properties. And we use that to design the compounds that we're going to prioritize, the new chemical syntheses that we're going to make, the new compounds we're going to explore at Iambic. But as I emphasized earlier, just doing AI isn't enough. You have to then convert that into chemical syntheses. You have to then convert that into high throughput biological assays. You have to actively learn on that, do all of the automated data analysis, do that in a high throughput clip. And that's something that we've really worked a lot at Iambic. It has a huge amount of engineering under the hood to really make that work at the pace of 1,000 molecules every week, where you go from new designs through chemical synthesis, through biological assays on a 7 to 10 day loop. So getting that answers to your questions at that clip and making the next iteration of discovery happen. Just one word or two about those key technologies. Neuroplexer is a co-folding model. There's others that you'll undoubtedly be aware of. AlphaFold won the Nobel Prize, rightly so, is with their breakthrough AlphaFold 2 technology. And Neuroplexer is a similar class of tool where it strives to predict the three-dimensional structure of a small molecule or a fragment of another molecule to a protein complex. The point there being is it can predict where the molecule binds, how it impacts the protein structure. Does it induce allostery? Is it likely to be a strong binder or a weak binder? Can you engineer selectivity? All of these questions are very useful in terms of structure-based design with a tool like this. And really, it takes out or it diminishes substantially the need for time-consuming crystallography and cryo-EM. And Neuroplexer is a powerful tool for doing that. We've shown this in open source benchmarking that in head-to-head competition versus AlphaFold 3, the most accurate method otherwise out there. It's more accurate in head-to-head competition. And furthermore, it has a very well-calibrated confidence metric that tells us not only what it thinks the structure is, but whether or not it's confident in that prediction. And for the 50% most confident, when it says it's right, it is indeed right. So this is really useful for guiding decision makings at Iambic. The way this was achieved was through a novel flow matching architecture. Others have used generative diffusion. We used a different architecture that allows us to get this better accuracy, reduce physics-based stereochemical errors, and to get that, improvements in accuracy, and to do that at much lower computational cost. AlphaFold 3, for example, will take this representative length of time on, these are expensive A100 GPUs that are harder to come by, and these are commodity data center GPUs. And you can see even on that comparison, it's over 20 times faster. So this allows us to predict the way molecules bind structurally, but furthermore, there's everything else that you have to get right. There's all the other dozens of properties that you have to get. Does it, have the right ADME properties, the right PK properties? Do you have the overall selectivity across the kinome or whatever portion of the protein space that you're working in? There's many different things that you have to simultaneously get right. And really, Enchant is our answer to that problem. It's a massive transformer model. It's trained on over 15 different shapes of data. So tabular data of all forms is one shape. 3D structure of all forms is one shape. Omics data, physics data in terms of quantum mechanical structure, unstructured biomedical text. So it's trained on over 7 trillion data points, and it's larger with every version. We've announced version 2. We'll have version 3 next year. And it's a truly massive foundation model that allows us to accurately predict across many different endpoints that are really relevant for the way in which we do drug discovery. So here's an illustration of that. In every data point that we were able to do head-to-head comparisons versus TX Gemma, DeepMind's fine-tuning of the Gemini model for drug discovery endpoints, you can see how Enchant stacks up. Here, Pearson R of 1 is perfect correlation between the predictions and perfectly held out experimental data. And you see it across different ADME, target-specific activity properties, pharmacodynamic properties, other physical chemical properties that are particularly challenging. And you can see that Enchant, has a very high degree of accuracy in being able to do that. And that's really core to how we design compounds. I want to convert, and I am mindful of time, so I want to convert to kind of the pipeline and the way that we do this. And I'll focus just on our lead program, the HER2 program. But it's the first of a series of clinical programs that we will have. The HER2 is already in clinic, as I mentioned, and we have two additional programs which will be entering clinic in 2026. One is KIF18A. This is quite a novel target related to chromosomal instability and a synthetic lethal mechanism for tumors related to the mitotic step of cell division, and CDK24, which is just an incredibly important a pair of targets that we're seeking to selectively inhibit with the eye towards addressing HR-positive, HER2-negative breast cancer population, which is a massive and underserved population. So these are our oncology-focused lead programs internally, but we also work in partnership with other companies. We've announced one with Lundbeck in terms of discovery collaboration in the migraine space, and I'm comfortable saying that we'll be doing more of those in the short term. In fact, Iambic has done actually 4 major partnerships in the last year, in addition to Lundbeck, which was inked just about a year ago, which, as I mentioned, is a discovery collaboration, meaning that we are doing internal discovery on a target of their interest, where they pay for everything and give us milestones and upfronts. We have a different partnership that we announced with Revolution Medicines. This is a pure technology partnership. So Revolution Medicines is not having us do any chemistry or any biology. They give us $25 million exclusively for one thing, and that is the neuroplexer tool. So we are letting them use neuroplexer. We're fine-tuning it on their in-house three-dimensional structures, and we're letting them use that. So it kind of illustrates the value creation in terms of the discovery engine alone, in terms of the technology engine alone. On the clinical side, we've announced a partnership with Jazz Pharmaceuticals, kind of where they are giving us $10 million of drug product, and we are combining that with our drug in patients for demonstration of the efficacy of that combination in breast cancer patients that are being treated today. In fact, with that combination, very exciting. And we'll have another announcement, I think, in forthcoming, which will be with a combination of both technology and a discovery collaboration. I want to talk a little bit about the HER2 program in the remaining couple minutes. And really, this is an illustration not only of it's the rubber hitting the road in terms of actually creating compounds that can make a difference in the clinic. HER2 is, of course, one of the most well-established oncogens, but it represents still an important unmet need with patients experiencing HER2-driven cancers across many different tumor types: breast, lung, colorectal, gastric, and the list goes on and on. This impacts a large number of patients every year. And the reality is that a large fraction of these patients, 40% plus in breast cancer and in lung cancer, the two largest indications, experience brain metastases over the course of their disease. The brain is a safe harbor for the tumor, and it will metastasize there, and that ultimately will drive morbidity for those patients. So this really informs what is needed. The ability to have a brain-penetrant drug is very important. The ability to have a TKI, a small molecule drug, a tyrosine kinase inhibitor, a pill, if you like, that can achieve inhibition of the HER2-driven cancer while avoiding extremely severe off-target toxicities. HER2 small molecule drugs have historically had punishing levels of rash, dose-limiting liver toxicity, and this is related to a lack of selectivity for HER2 versus closely related proteins like EGFR, which drive those toxicities. And then thirdly, in addition to widening that therapeutic window, broadening that population through the treatment of brain metastases, we also want to be able to widen the patient population by treating not just HER2 wild-type driven cancers, but also those cancers that are driven by HER2 mutations. There's many oncogenic mutations of HER2 which further drive different forms of cancer. So we want to have one drug that achieves all three of those aims. And really excitingly, we've been able to do that with IM 1363. This is a very novel scaffold, chemical scaffold. We have shown that it binds HER2 uniquely among all known HER2 inhibitors, 20 plus years of research. It's the first to ever bind in the inactive conformation of HER2. All others bind in the less thermodynamically prevalent active conformation. So it binds the inactive conformation, confers these very differentiated properties in terms of extremely high selectivity to maximize the therapeutic window, extremely broad pan-mutant activity, and also excellent brain penetrance. And it's progressing very well through clinic, completed dose escalation. I'll show you some of the data we're excited about, which has been reported externally at ESMO just recently. And we're open at 30 plus sites, going to 60 sites over the course of the next year and a half across Europe, EU, and we're also expanding at a number of Asian sites. Just one illustration preclinically of why we're excited about this. The best-in-class drug outside of IM 1363 is Boehringer Ingelheim's zongertinib. This is the indication that they have focused on, non-small cell lung cancer with mutations. So in Garanib, this is in a mouse model, but this is at an exposure that is higher than what they're able to safely dose in humans and carry forward in humans. And these are IM 1363 doses that are well tolerated at the corresponding exposures in humans. So Garanib is blue, it does not lead to tumor reduction. IM 1363 is red, it leads to deep tumor reduction at multiple doses here. And this is, what you want to see. I bring this up because it illustrates one of the really unique properties of IM 1363. IM 1363 targets the tumor tissue in almost a singular way. Most drugs show what zongertinib shows. This is the drug in the plasma, this is the drug in the tumor at two different time points. There's much more drug in the plasma, which means, floating throughout the body, causing peripheral toxicity, than in the tumor, where it's actually doing the work of killing the tumor. IM 1363 shows the exact opposite. It shows vastly more drug in the tumor than in the plasma. And in fact, at trough, after 24 hours post-dosing, it has more drug in the tumor than it has at any point, even at the peak in terms of concentration in the plasma. So this is a really magnifying effect in terms of the safety of the drug. We've shown that this also happens in humans. Now, these are human biopsies, and we see the same thing. This is the drug in the plasma in humans. This is in the tumor biopsy, the concentration in the tumor. We see this again at a higher dose. The time course for the drug in the plasma, at 27 hours afterwards, it's gone. It's like the patient doesn't have any of the drug in their system, except in the tumor, where it's doing that anti-tumor effect. So this is a really unique feature of the scaffold that we've arrived at and a potential differentiator. It leads to exciting activity that we're seeing and we're gathering data on. This is an example in there's no approved HER2 small molecule drug for this form of lung cancer. We're very excited at the depth of the responses that we see here. This patient showed over 90% tumor reduction. We've seen it in other areas. Gastric cancer also has no HER2 approved drug. Excellent and rapid responses in terms of the tumor reduction that we're seeing in these patients. So we're excited about that. And at ESMO, we were able to show a compilation of data that we've gathered as of the cutoff date, and we continue to gather, of course. And really the upshot here is that we have a broadly active and broadly well-tolerated drug that's very encouraging for future development. To dig into that just one click further, these are very heavily pretreated patients. These are patients that have had, we're listing the prior lines of therapy, 6 lines of therapy, 5 lines of prior therapy. These, for those who are familiar with ADCs, over half of these patients have had Enhertu, which is the X on all these things. These are heavily pretreated patients that are, have exhausted therapeutic options. That's why they're in oncology phase 1. And what are we seeing? We're seeing that we have, a very high fraction of responses in such a late-stage patient population. We're seeing broad activity with the responses including non-small cell lung cancer amplified, ovarian cancer amplified, breast cancer with a brain tumor. This is actually the brain tumor of the breast cancer patient. Non-small cell lung cancer mutation. It's a different patient population. Gastric cancer, renal cell carcinoma with a mutation. So these are very distinct. All of these are responses. All of these are different forms of cancer. And in almost all cases, there's no HER2 approved small molecule drug. So we're very excited about that. And in fact, we're also have shown that there is no evidence of significant rash or liver toxicity, the primary dose-limiting toxins of other HER2-driven drugs. So that's another important thing. We're seeing broad activity, we're seeing differentiated safety, and we're excited about the potentials to combine this and to apply it as a monotherapy, and we're aggressively carrying that forward even as we bring other drugs to clinic. So with that, I will conclude. I'm very happy to thank the team, thank David and Kevin for the chance to speak to you and to address questions in the meantime.
Thank you. Yeah, what an amazing journey. One thing I wanted to start out by asking, because I know there's some attendees that uh, are candidly researchers that are probably thinking about doing this. So I always like to start out by almost the birth of what was Entos at the time and has now become Iambic. So when you were at Caltech in your lab, you had collaborated with Fred for a while. What was the what was the thinking? What was the motivation? What was you know,
Lots of
yeah,
yeah.
just walk us through hey, I'm going to take the leap now, and I'm going to actually take this out. And I do remember, I mean, at the time you were unsure, and I won't disclose the names, but there were basically Fortune 500 companies that you were working with that were non-biopharma
Mhm.
to kind of validate some other potential paths that could have been quite interesting as well. And there's a lot of optionality there. So just walk us through getting it out of the lab, getting it started, why you made that decision and that journey.
Well, I'm going to be very, thanks for that question. I would say that it was absolutely a pull mechanism as opposed to a push mechanism. I loved Caltech. I loved my time there. I had just a magical 15, 14 years of my career there. It was the place where I grew up. And, there was a version of my future where I stayed there extremely happily and continued to do the sort of research that I had been doing to date. And I think what I increasingly saw in my own personal journey was kind of a an appetite to participate in the translation of the science to to the ultimate impacts of that. I think that at least in the way, and people do this differently, and this can be done in academics. So I don't want to say that the journey that I've taken is by any means the right one. But at least the style of science that I tended to do was telling the first chapter of a story, saying this is in principle going to help this and that, and then kind of handing it off. And I increasingly, and I know my co-founder Fred increasingly wanted to see that through and kind of live that journey of the technology. And entrepreneurialism is a great mechanism for doing that. And and Caltech was very supportive of me exploring that possibility and ultimately making that transition to pursuing that possibility.
Yeah. And then, walk us through a little bit in terms of, so then you decide or you see this, you know, potential opportunity to apply this in the therapeutic space. Just what, and then, just for those aren't who are, you know, tuning in, it's become in essence an oncology focused franchise to date, at least with your owned assets. So why is it going into oncology, which obviously probably all of us have lost dear friends and people to that. So it's very important. But like why'd you go in that direction? And then maybe walk us through how you're thinking about the partnerships, which tend to be in non-onc spaces. Yeah.
No, I think that's exactly right. And I'll take those two sides of the question in turn. As I as we discussed a little bit earlier, the original platform was very agnostic to the domain of application, right? It was a very flexible, if you have a molecule, this will tell you the properties of the molecule kind of platform. And that could have gone in many different directions. And in my academic work and in other projects, I collaborated in many different directions that way. And in fact, it worked in batteries, and one of the endpoints of that is a fantastic fluoride ion technology that became Flyon, for example, as another startup company that I'm less involved with, but that nonetheless was related to some of the underlying science. And I think that so why oncology, why drug discovery? I think that it became very clear very quickly that if you have a tool, you have to have a direct line towards a commercializable product, so that it can not only validate the tool, but that it can, just have a line of sight towards not just having promise, but having profit. And I think that's really important. And as we looked at the timescales for different places, small molecule drug discovery had a clear timeline. It had a clear progression that we thought we could shorten, and it was very well aligned with the science. So kind of the first step was kind of narrowing it from all chemical applications to small molecule drug discovery. And that was an alignment of kind of clarity of a commercial journey, in combination with compatibility with the science. Within therapeutics, generally, I mean, there's many, huge opportunities and unmet needs for patients in terms of developing new drugs. Oncology was another very particular choice. There is a huge amount of unmet need, but there's unmet need in many disease areas, right? The particular thing about oncology that was important for us was that in phase one in oncology, as I just showed, you're treating patients with disease. In every other disease area, you are not treating patients with disease. You're treating patients you're treating healthy volunteers. So at this point in time that Iambic is at, what we were able to say is that we have both a safe and an active drug.
Hmm.
And that's very validating for everything that we've been doing up to this point. If I had made a different choice about therapeutic area, if we had, we would be at this point in time, but we would only be able to say it's safe and it has exposure, which is great, but it doesn't show that it actually does the disease work. Yeah.
Yeah.
So for that reason, it gave us the opportunity to validate and de-risk at an earlier time point than other choices would have given us. And that was, I think, being an entrepreneur, it's like being an untenured professor. It's like it's just a race against time to be able to show you that you can deliver. And this was part of the choice to make that happen. And having done that now, we can now do that in other therapeutic areas. We have internal programs in other therapeutic areas, and as you're correctly pointing out, we do it with partners in other therapeutic areas as well.
Yeah. One of the things that I distinctly remember this uh, last year when I visited you guys at your office. And Kevin, I think we're getting a little bit of feedback from your machine. Um, is that you said that the with the dosing, the AI actually the using your machine learning, you can actually be much more kind of aggressive, although it's not actually technically aggressive because computationally you can predict it that you can actually dose much higher and not feel that the safety is being compromised. Can you just walk us through? Because I think sometimes people are like, oh yeah, AI is helping you identify things, but it goes much beyond that, actually. It's like a core part of what you do during the entire development cycle, even when it's in clinical trials, like it doesn't stop. It's like letting you optimize dosing and that's why your readouts are so, I mean, kind of impressive.
Thank
Can you
you.
maybe walk us through how you continue to use it along the way?
Yeah, that's I think that's a it's a couple of different questions that are, I think, really timely for the field, in fact. On the one hand, yes, we can use AI to prioritize which molecules to make and which experiments to do so that we can assemble the data package to bring to the FDA as fast as possible. That's, we've definitely used it in that way. Another thing that we do now, and this has been part of the opportunity that's been created with Enchant, is we can make reliable predictions about how drugs will behave in early stage clinical readouts, but we can do that before very early in a project now. So we can say, what is the likely human exposure of this drug? Is it going to get to the right levels? What is the likely clearance rate? What is the likely on-target engagement? We can make statements about those things very early in a program. For before we even synthesize the molecule, we can make
Mhm.
predictions on this. And that allows us to prioritize chemical families and directions of the program in an early stage when you can still make choices like that, that will ultimately translate to higher probability of success. I think that proof of the pudding will be in as we bring in programs 2, 3, and 4 and show that they continue to show the same success of our first program. But in all of the, benchmark versions of that exercise, we see that that is a real opportunity that we're able to take advantage of, and we're actively doing so. I think there's another interesting set of questions about now that we're in clinic, can tools like Enchant help us to prioritize cohort A over cohort B? Can it help us to stratify patients? Can it help us to identify biomarkers that are going to be more likely to respond, for patients that are going to be more likely to respond? Can it ultimately be used for diagnostics and prognostics about which therapeutic to apply? And we really firmly believe the answer is yes. We think that the ability to have a massive transformer model that connects, sequence data, biological target data, therapeutic molecular property specific data, it will begin to erode the difference between therapeutics as a field and diagnostics as a field and biomarker development as a field. It'll erode those things and have one mechanism for correctly answering that question across a patient's journey. And we're very excited about that and think that that's, going to be part of the future chapters of this technology in the company.
Hey, David, how's the feedback?
It's fine.
Okay now? Yeah, so if I could, I'll jump in here for a second. And we're getting a number of questions, David. Maybe if you want to take a look through those while I ask. What I think is a great, what you just covered is a great lead in, to what was a pretty, dramatic announcement in April of this year by the FDA to basically phase out animal testing for many drug types and really start to focus more on in silico methodologies and human-based lab models. So that presumably bodes really well, for you and for Iambic, for its investors, and for patients with these, unmet medical needs. So maybe you could just, talk about that a little bit in terms of its potential impact on the industry and Iambic's, role and how the company's now positioned as this starts to unfold.
Yeah. No, I think that's definitely a very concrete illustration of how the FDA is thinking and moving in this direction. And you're right, Macri and the FDA announced the plan to begin using in silico predictions as a supplement to animal data to reduce the amount of that data that would be potentially needed. That is a really big shift to the extent that that starts to get used.
Right.
Historically, AI is a great way to figure out what experimental data package to present, but they were only looking at the data package, right? And they weren't looking at any
Right.
predict. Um, you know, Interestingly, the FDA uses modeling on their side to determine what is a safe initial dose, to use what is a safe, safety margin or what is an appropriate level of off-target risk and that kind of thing. So they are using modeling on their side. So I think that this is kind of an invitation to, not just use modeling on their side, but to use modeling on the therapeutic developer side and to start to have those two things start to meld together. Our answer to that again is Enchant. We think that that's a great tool for helping to bridge that boundary. We're talking with a number of different parties about the right way to, expand the footprint of enchant across not just therapeutic development, but other applications. And I think that this is going to be the trend of the field. And we are excited about where we sit in that.
And Tom, just as a thank you, and that's great. Just as a follow-up to that, maybe just to remind everybody why, speed is so important here and why it's so impressive that you got, your first product into clinical trials in 2 years, when the average is 5 to 6 years. A well-established, empirical, average time. What's so important about timing, and what are the considerations? How does big pharma think about this? In terms of each day of development, right? I think so where I'm headed with this. So let me just hand it back over to you.
I think that's right. Time is just, it's a very clear measurable, right? But it speaks to so many other important things. The burn rate of a big pharma company or of any given project is extraordinary. So time translates massively to cost. A faster time implies that you have gone down fewer incorrect roads and had to backtrack, each of which is extremely costly and financially in terms of the competitive landscape. Oncology is a brutally competitive area. There's always many companies that are working to race to the
Hmm.
right target, and time's a big element of that. I do want to say that time is only one part of it. Time is important, cost is important, but at the end of the day, really having a differentiated drug is paramount. Iambic does not strive to be, just a fast follower company where we're gonna outrace other people with a Me Too drug. Having that
Right,
real differentiation of the drug is the way to get durable approvals, the way to get, to really move the needle for patients and to do that in a long-term way. And if you work in an area like HER2, the bar is higher to achieve differentiation, right, than an area where
right, right,
but you still need to achieve that differentiation to achieve that. In the eyes of pharma,
right,
yeah, they want to participate in that speed. They want that breadth of applicability. But they also, I think, look at Iambic's ability to create real drugs, deliver them to clinic, show that they're actually working in clinic as just a meaningful validation of everything under the hood. It's hard to diligence AI. It's hard to know
right.
whether this tool is better than that tool and to look at slide decks and benchmarking. I mean, it's just hard. So, if they like what they see coming off and they can quantify that,
Mhm.
that just speaks volumes about what's going on under the hood. And I think that's an important part.
And then just one final thought on this particular topic is just to frame that in terms of the patent dynamics that a typical company like Iambic or any biotech or biopharma is dealing with.
Yeah, patent dynamics are very interesting. I mean, so there's patent cliffs inevitably. There's differences between the way small molecule drugs and antibodies are treated. How long that will remain the case is an open question. But nonetheless, any way you slice it, the faster you can get from exploration or discovery of the chemical matter to the ultimate journey through clinic and commercialization, that adds critically important years to the patent coverage and the ultimate value of the program itself. There's also very interesting questions about, the challenges of once you patent, then there is other opportunities for other companies and other
Right,
people to try and patent bust. So having that speed on the back of that is an important thing. And AI is also going to play a role in designing patents for maximum protection and thoughtful ways there. So all AI will infect everything, and I think that drug discovery is such a fascinating field because there are so many interdisciplinary challenges that can be made more efficient and better through technology advances. And that's something to lean into. The opportunity to really use technology to make better medicines, to bring those to patients is just a fascinating multi-dimensional multidisciplinary
right,
problem that
right.
very rich to work on.
Right. And just to put that quickly in context, some drugs can take, 12 to 13 years to actually develop from inception to commercial launch. And assume a typical 20-year patent time frame, then that doesn't leave a lot of exclusivity left to really, earn all the money back that was invested in the development. You're on path to hit commercialization potentially in the 6 to 7 year
Yeah.
time frame,
Yeah.
which is, rough numbers, half of the time, leaving, significantly more patent life to a product once it's launched.
Yeah. And that back end of the patent life is when you've already reached your peak sales and it's really
Yeah.
that's
Right.
you're really
That's right.
benefiting from it. Yeah. So it's
That's right.
that year matters. Yeah, that's really true.
Um, Switching gears or just maybe picking back up on the partnership side, and there's I think one of the questions is about this, just what's stopping some of the larger biotechs and biopharmas from doing this type of thing in-house? And I did want to I guess clarify that my understanding is that, or previously we discussed that you like when you're doing partnerships, I believe you only do them in non-oncology, and then you also uh, only use the previous versions of your platform because you want to keep your most up-to-date version to create your own assets. But correct me if I'm wrong on those, but then also maybe
I'll
explain.
push back on both of those. Yeah.
Okay. Yeah, yeah, yeah. Awesome.
Historically, we have only done the only one we've announced is on a non-oncology target, namely a migraine target. We don't necessarily see a problem. We do you have to be very thoughtful. Whenever you work with a partner and you work on a target, they get some level of exclusivity around that. We sometimes people do that across broad therapeutic areas, which we were not interested in doing, just because we have a platform that can move in a lot of different ways, and we want to be able to, take advantage of those things that we don't even foresee doing today. Um, we do not prohibit there are plenty of oncology targets that we are not going to work on at Iambic. We know that. And in many cases we've evaluated them and decided that, for our own dime, it's just not there's just not the thing that's going to rise to the level in terms of what we want to do. So, we're very happy if somebody else wants to work on that and they want to pay us and they want to, cover all the costs and they want to bring in all the expertise and make us stronger at this particular strategy in the meantime and give us additional data in the meantime so that our machine learning models
Mhm.
get better. We will do those targets, and that can be the right thing to do. So we are open to that kind of thing. I think around the technology, Iambic has generally not distributed technology externally. We have largely kept Neural Plexer in-house, particularly with the advanced versions. We've never, disclosed Enchant externally. The Revolution Medicines deal was a departure from that, where we got very good economics around just a very specific use case of one of our machine learning tools. So I think it's part of Iambic's ongoing journey to balance the kind of productization of the platform for
Mhm.
others, even as we do it for ourselves. I think that the right choices that we make at a given time is a little bit dependent on the stage of the technology and the stage of the field. Co-folding as a technology field is a little bit different stage than transformer models that are more broadly applicable. So the right answer, even across models, will be a little bit different. So we take that on a case-by-case basis, but we are extremely mindful of protecting our own competitive advantage, even as we realize that these tools are hungry for data. They're
Mhm.
extremely broadly applicable. There is something crazy about having a tool that could work across almost any drug product and then use it for your own tiny three-program biotech, right? That's a mismatch. So there have to be ways to share that don't disadvantage us, but then, help to realize the value.
Yeah. Well, and not without not getting too specific on the economics, but it can be quite lucrative and with the expense, there's still the expense as you go into clinical trials, right? Like that hasn't dissipated. You can be much more efficient and
Oh
thoughtful in
yeah.
how you do it, but there's still, significant costs involved. But it's been interesting to see how, and what I call engine two, which is the partnership route, can feed into and dramatically reduce the amount of external capital that you need.
Yeah, yeah. I think that's exactly right. Every partnership that we do adds quarters of runway to the burn of the company that is meaningful in terms of the fact that we can reach different clinical inflection points for the main program. So the platform really does drive not only the ability to make our internal drugs, but it is a provides revenue that allows us to extend that runway. And technology is scalable in the way that drug programs are not, right? We can't have it's not practical for a company of our size to have a dozen programs in clinic, right? We're only going to have a smaller number, but we can make a dozen or 2 dozen technology deals because that scales. So those
Yeah.
are reasons to take advantage of both sides of the coin.
Yeah. Um, there's a few questions. Let me just kind of hit on the technology itself, maybe. Let me just hit a few of these. And can you t tell us more about the multimodality signals you use to train Enchant? You mentioned leveraging in-house 3D structures from partners. Are they
Yeah.
X-ray crystallography or cryo-EM? And
Yeah.
then there's one about external data, and maybe even unstructured data.
Yep.
Um, so just curious about some of those things. If you could maybe just comment on deeper dive on the technology itself.
Yeah. For the asker of that question, I would invite you to look at our ICML paper on Enchant. You can find that by way of our website. So we did describe in a publication an early version of the version 1 of Enchant that we've been building on since then. But I think it'll give you a sense of a lot of those kind of core questions. We do both structured data and unstructured data. It includes three-dimensional structures of cryo-EMs as well as basically anything in the PDB, as well as tabular omics data in other forms. Much of that data is public data, but we also have large repositories of internal data, both experimental and synthetic computational data that we've created through quantum chemistry calculations as well that we use internally as well. So that's, in terms of just a technology challenge, a lot of it is, creating ways for the transformer to work on different data shapes. So kind of creating those modalities for input into the transformer, having those be tailored to the real physics and real biology of drug discovery has been a lot of the kind of innovation that happens in terms of building the transformer model. And you can read about that in the ICML paper as a good starting point.
Gotcha. So I posted your website in the chat if people want to start to explore some of the postings that you've done and the papers that you guys have put there. Um, in terms of there's a question on maybe looking out a few years, just like what's the this guest used the word end game, but just like maybe what's if you had to look in your crystal ball for Iambic, what's your design? What company are you building?
Yeah.
Maybe that's the best way to put that. That's a very founder specific question. Yeah.
Yeah, we are pursuing the vision that better technology innovation can lead to better medicines, first-in-class drugs, best-in-class drugs for patients. That is the journey that we're on. That's not a one drug process. That is a repeat, that is a ongoing innovation process. And so we as a company are building for the long term. We're thinking about this as the long term. There's no stage of clinic beyond which we will not go. We are not taking commercialization off the table. We are building a company for the
Mhm.
long haul. We will make the right choices in terms of partnerships that make sense on a given program and the given stage of the company. And in terms of end games, of course, we have our obligations to shareholders in terms of making sure that they see that return in various forms. But we are building this company for the long haul and definitely thinking in that way.
Yeah. Yeah.
I can't quit tenure at Caltech and then flip the company in 2 years. I wouldn't have anything to do with myself. So
Except create your own lab, right? Yeah. Yeah. Um, no, in terms of I appreciate that, and I remember I fondly remember the chats we had in your lab at Caltech about real. I was struck by the ambition of what you outlined, and almost the energy maybe is the word I'd use around energy and frustration around how biopharmas have not maybe been as responsive as they could be to some of the uh, diseases that are out there, and a desire to really change that. And so I think that mission, I know that has continued to energize the team, will serve you well in doing the right thing and building a long-term durable franchise.
yeah, thank you for that,
So
David.
yeah,
It's an exciting journey. We're incredibly lucky with the team that we've managed to recruit around the company. And it's scientifically it's fascinating, and yeah, in all ways it's fascinating and rewarding. It's been great.
yeah. Um, have
So
you? Oh, go ahead, Kevin.
just one other question for you, Tom. This has been great, by the way. If somebody were to take a look at your website and look at the board and then look at the scientific and clinical advisory board, it's just really stacked. I think by any estimation, names that are well known in the space, which presumably requires a fair amount of effort to recruit them, and then to engage with them. So. How are you managing that? Because it's a big group and again, an impressive group. And are you getting sort of the ROI from that in terms of the time that you and your team need to invest in managing that in terms of what you're getting back from them? And what's your advice to other, existing and would-be founders as they think about, how and when to sort of build that out?
I think that putting experienced people around the company in the early days is incredibly valuable. We were really fortunate at Iambic to have, for example, Francis Arnold be among the very earliest scientific advisory board members. These people have seen it more than I had seen it. These people know people that ultimately are helpful in terms of they might not know the answer to every question, but they will help you to learn the right person to help ask the right question to inform your journey. And similarly with our first key board members as well, they help you to navigate. Every first-time entrepreneur is on a journey, but just the amount of stuff you don't know is just staggering, right? You don't know what the words mean. You don't know what stage you're supposed to do which stuff. It's staggering. And you do need shepherds and stage appropriate shepherds through that overall process. And it's ongoing. You're just
Right.
at that stage always because you're always in the tip of the spear of learning. And and those people help you and building that group around the company is absolutely important.
Any?
I do have we should probably I can't go too I have a
Yeah, yeah, yeah,
a big problem waiting on the hour in fact, so I have to go.
yeah, yeah. Any I don't know, any lessons that you've learned that you want to impart that you're like, yeah, that was interesting, or yeah, just takeaways for the first 6 years now of, because I think technically it was probably 2019 when you started to, yeah, I think we met in 2019, maybe
Yeah, no, yeah, we did the seed round in 2020. We started to get serious about that intention late 2019. Any major lessons? Um, I mean, I think that you have to enjoy the process. I think that's just an important thing. I think that there's challenges, there's good days, there's bad days. Sometimes days have both elements.
Mhm.
There's a lot of just it's surprisingly similar to academics in the sense that you have many more failures than you have successes. You need to lean into those successes. You need to turn failures into successes by seeing the bright side of what's going on. You need to have optimism. You need a core thesis that has merit, that you believe in that is meritorious objectively, and you need to follow that. And so long as those things hold together, you're working towards something that is legitimately worthwhile, and you maintain that optimism in the mission. There's a way to get there. And yeah, I think that's what I've found.
That sounds, yeah, some wise words. Yeah, we tell the scientific founders to be, find something that you're insatiably curious about.
Yeah, that's good.
Well, thank you so much for carving out an hour of your time. Really appreciate it. And thanks to everyone that joined us today. And have a good rest of the week and a good holiday season, everybody. We'll conclude there.
Thank
Thanks,
you, Dave.
Tom.
Take care.
Yeah.
Bye.
Thanks, Tom.
See you.